Hello, I'm David Ishii. I'm a biohacker. This is about a DIY coronavirus vaccine that me and some biohacker friends of mine made. Um, as you can see, we're here in my lab. It's really just a shed that is a fully functioning genetic engineering lab. So, DIY coronavirus vaccine. This all started in May of 2020. Obviously, coronavirus was a global pandemic at the time. A little more introduction about me. I started out as a dog breeder. This is how I got into biohacking. Biohacking is like genetic engineering from a hacker's ethos. So I started out from dogs. I'm a dog breeder. I breed working mastiffs, and I try to make sort of genetically improved working mastiffs. And I've made a lot of progress over the last 10 years. That's what got me into biohacking. Biohacking is just genetic engineering, but from a hacker's perspective. It's about freedom of information. It's about accessibility. It's about making things in a way that doesn't require a big institution, doesn't require millions of dollars. Everything from, making genetically modified dog sperm to fun things like making bioluminescent jack-o'-lanterns. And those projects, my main project is on making genetically enhanced dogs that are healthier, longer lived, better at doing their jobs, that sort of thing. But I've also done some work on human gene therapy. And so that's kind of how I got involved with this project, because around May 20th, even though the date for some reason says August 14th now, I guess they updated it. But anyway, it was May 20th. I wrote it down. There was a paper that came out about a DNA vaccine for coronavirus, SARS-CoV-2, that was studied in rhesus macaques. And so they're monkeys and they're model organisms. And the nice thing about a DNA vaccine is it's very, very close to a lot of the work that various biohackers have done on genetic engineering, like gene therapy stuff, because DNA is easier than viruses, right? And so without having a big, I'm not going to get too deep into it because I know a lot of people here probably won't have a great background in biology, but basically a vaccine just presents some part of the virus or bacteria or whatever it is that you're trying to make a vaccine against to the immune system, so the immune system can see it and respond to it and develop immunity, usually in the form of reactive T cells and antibodies. So a DNA vaccine, instead of having something like a killed virus or a virus that's grown in a way or genetically manipulated in a way that makes it unable to replicate or weakened, a DNA virus takes one particular gene from a virus or bacteria, really, but any organism that you're trying to make the vaccine against, and takes the DNA itself, puts it in a vector, in this case a plasmid, which is just a little loop of DNA, and then that gene is put into the person or animal that you're trying to vaccinate in a way that it actually enters their cells so that it genetically modifies that organism in a very small way. Just a few hundred cells around the injection site get modified so that they produce one of the proteins of the virus. And in this case, it's the spike protein. And the spike protein is a good candidate because it's a large external protein. It's on the outside of the coronavirus, and it's used to actually bind to the human cell receptors that it recognizes, and then that's how it knows to enter the cell. So what we did is we took the DNA from coronavirus. Now, coronavirus itself doesn't have DNA, it has RNA, but luckily you can translate it pretty easily. So we took really what was the protein sequence, and we converted that to a DNA sequence. And then that's much more doable because you never actually need the virus. You never have to have an actual sample of coronavirus. All you need is the data. It's just information. So all you have is just a sequence of amino acids, which you translate to a sequence of nucleic acids, a sequence of DNA letters. And then you can take that and use, and everything is done digitally in the design phase, right? So there's never a need to actually have a sample yourself, right? All that can be handled in like biosafety labs where you don't have to worry about it. So in this study, they did rhesus macaques, and here are their results. I'm not going to go through it really, but they tested various versions of this thing. So they had they tried the spike protein, they tried pieces of the spike protein, they tried spike protein pieces with modifications, and they compared all the results. And what they found was the macaques who had just the whole spike protein had the best results and were protected from the virus. They actually tried to infect them afterward. The control group got sick. The vaccinated monkeys did not get sick. So their DNA vaccine worked. That's when me and two friends of mine, Josiah and Daria, also biohackers. Josiah is a biohacker from San Francisco, Oakland, really, and Daria is a biohacker in Ukraine. And both of them have their own biolabs, just like I do. They both do genetic engineering in their own projects, and we all kind of decided to get together and see if we couldn't replicate this project in humans, because that's the logical next step for this thing. Is if humans, or if it works in monkeys, there's a decent chance it'll work in humans. And instead of spending months and months worrying about FDA paperwork and clinical trials, because we're biohackers, we can just try it. We can just make the stuff ourselves, get the stuff ourselves, and just do it. So that's what we did. Let's see. So this is the vector that we designed. It's heavily based on the paper, the spike protein. So this is a plasmid map, by the way. So this represents the vector. This represents the little loop of DNA. So if I were to zoom in on this, it would actually be a series of genetic letters just all the way around the circle, about 10,000 of them. And so so what you find is that this section is the spike protein. Here's a promoter that tells your cells to make the spike protein, or terminator, and all of the rest of this is used in replicating it in bacteria. This is the part that's active in the human cells. This part technically is too, but don't worry about it. It's just background stuff. It's sort of leftover programming. And so the part that's meaningful for our thing is right here. And basically all it does is if it gets in a human cell, the human cell will read this DNA and do what it tells it to, which is to make this spike protein. And when it makes that spike protein, it'll present it on the outside of the cell. And then your immune system can notice, hey, this is not a human protein. Something's up with this cell. It'll take it back to your lymph nodes, and the B cells there can start working on it, and they can start trying to come up with antibodies that stick to this thing, because that's what they do. Let's see. So the first thing we needed to do was we needed to order it, and we did. We ordered it from we had it manufactured synthetically. So you don't actually have to make DNA in organisms anymore. You can make DNA just chemically. As long as you know the sequence, you can have it synthetically manufactured. So you just send the people at these labs. We used GenScript. We just sent them money and a DNA sequence, and they sent us a tube full of DNA. And so that DNA then becomes, that's the vaccine itself. And the nice thing is, it's not living. It can't replicate. It can't spread to other people. There's nothing it can really do except make spike protein if you put it in a human cell, or really any mammal cell. But the spike protein, we needed to test that it works. So first thing we did was we did some mammalian cell culture. So you can see here some of the culture flasks, and inside these flasks and inside my chicken incubator, human cells are growing, and we genetically modified those cells. So you can see this is what the cells look like under the microscope. And this is one of the modified ones. Now this has been modified with a fluorescent protein just to evaluate the protocol. But some of them were also modified with the spike protein. So we took the vaccine, put it into these cells, extracted from that the stuff that comes out, what it makes, and then we were able to test and show that we had spike protein. And so we collected a lot of our blood, the serum specifically. So when you centrifuge blood, you can separate the liquid parts from the cells. And so we saved a lot of this from before we took our injections and after. We even went and got we got our antibodies tested by third-party labs and those sorts of things. We also had labs replicate all the things that we did. So we tested for spike protein ourselves, but we also had another lab do it just to confirm our results. Let's see. And then it was injection time. So the injection, it's a large volume. We did exactly what they did with the macaques. So it was 5 milligrams at 1 milligram per milliliter. So that means 5 milliliters total per injection. So this kind of injection is similar to the one that Moderna is developing right now, except theirs is based on RNA instead of DNA, but it's basically the same thing. But the DNA has to be injected twice. So you do 1 dose, and then a couple of weeks later, you do a second dose. The first dose kind of primes your immune system. It learns to recognize the thing. It has time to develop a population of T and B cells that recognize it. The second dose is like a booster, and it really ups the amount of antibodies and stuff that are used in that. And then you end up with all 3 of us, we decided to live stream it. We did the whole thing as a class. So we live streamed every step, all of our results, all of our testing, all those sorts of things, so that everybody could sort of look over our shoulders and see how we did it and what we did and why we did it. And so this was injection day. And so that syringe, you can see, is 2.5 mLs. That went in one of my shoulders, and then another 2.5 mLs in the other shoulder that day. And then 2 weeks later, I did it again. That's a big injection. That hurt. So but it was still a lot of fun, and it was interesting. We all 3 kind of had like general fluishness, like you felt like you just got a flu shot kind of thing. The next day or 2, and then we got fine. And 2 weeks later, we did it again. We continued to collect blood throughout the process, and we continued to test the results throughout the process. And then we end up, and so this is some of our testing. We did ELISA testing, which I'm not going to get into, but basically you can tell by the color how much of a thing is in there. The stronger the color, which you can read by a spectrophotometer, the more of that thing there is. And we also did something called a pseudovirus a surrogate virus neutralizing assay, which is interesting if you want to Google that, but basically it can detect whether you have antibodies that will interfere with the spike protein's ability to bind to the human cell receptor that it binds to. So basically, if you have antibodies that can stop coronavirus from sticking to and recognizing human cells, then these wells will not have color. And if they do have color, then the virus is able to infect. And the stronger the color, the more infectious, and the less color, the less infectious. So as we continue to do it, this is how we were evaluating the results. This is a standard kit. You can order it online, and this is the same kind of thing they run in labs when they're testing you for anything, really, but in this case, coronavirus. And these were our results. Now, the results were really interesting because, like, this yellow line represents, I don't know if you can see my mouse, but the yellow line represents the injection days, right? So here's injection day 1, here's injection day 2, and the height of the line represents what's considered by that kit to be a positive signal. So below the line is positive, above the line is negative. So you can see we did the first injection, and this is Josiah's in blue, I'm in red, and Daria's in green. And interestingly, like, Daria already had some ability to neutralize. So she had some antibodies against it before we even started. Josiah had some background antibodies that we were able to detect with some of our other tests, but they didn't neutralize the virus. They just stuck to it. And that's one of the reasons this test is important, so that we can differentiate between antibodies that neutralize the virus and antibodies that just stick to it. So, but as you can see, all 3 of us, after the second dose, sort of cross that threshold between a negative result and a positive result. So we all 3 developed coronavirus neutralizing antibodies at the end of the work, and so effectively it worked, which we weren't necessarily expecting. But we did record everything on YouTube and stuff if you want to see that. It was an interesting project. I think it was intended to be educational, but the overall goal really was to try and share this information. We're not distributing this to anybody. We're not, which I mean, you can get the plasmid, but we're not like giving out doses of it. Like you can't go get, we're not going to sell you a vial of it or anything. That would be illegal, but the goal is to make this information available, because when DNA vaccines were first researched, they were heavily researched by things like DARPA because they're so easy to do. The whole project, from we're talking about it, we read the paper, to we've all got neutralizing antibodies, took like 129 days, something like that, 123 days. Very, very fast. And three people, which admittedly three people with their own genetic engineering labs, but three people with like a regular person's budget and, you know, a little time. And we didn't actually have to do all the testing we did. We did quite a bit of testing before we did the injection. If we had just gotten the DNA and done the injection, it would have been a lot faster. But we wanted to do a lot of sort of quality control. So the importance, I think, though, is that when you have global pandemics like this, you know, it really sort of illuminates the fact that we have very poor biosecurity nationally or really globally. If you know, I'm not in the camp that thinks this was a bioweapon, because it's not nearly terrible enough. If this thing had been a bioweapon, it could have had its mortality rate could have been 65, 80%. If you had the same infectivity and a much higher mortality rate, like the avian flu, for example, it was bad at spreading, but its mortality rate was 65%. Rabies is 100%. So there are much, much deadlier viruses out there. And if you were going to make a bioweapon, you could make something that would really be really, truly terrible, or at least not a individual. Like obviously, this is probably some of the most advanced stuff biohackers are doing, but I couldn't make a bioweapon. But a state could, right? North Korea could, Russia could, the United States could. They have the tools and the means to do that. And so if a state wanted to make a bioweapon, they could. And if they did and released it and it was really terrible, I don't think there's anything we could really do about it, because we don't have any kind of significant biosecurity, not on a national level or a global level. Just about no one was able to stop this. Some of the island nations had a better shot at it just because of the sort of nature of geography. But for the most part, this if there will come a time when this technology is used in sort of a bioweapons capacity, and our large, slow, centralized approach to medicine is just a giant, especially when it's heavily impacted by politics, is just a giant, slow, terrible machine that can produce things, but it's not flexible. And if that machine is the target, so if the first place that gets hit by this bioweapon is like the CDC, what do we do? You know, and I think it's important to have a distributed set of people with labs, with skills, with access to reagents and techniques and technology. Like I said, the DARPA was really investigating this technology as something that was super easy to make, super easy to sort of roll out in a disaster scenario, because it's so easy to make DNA. Like once you have a plasmid, you just put it in bacteria and let the bacteria replicate and then extract the DNA from them. And like E. coli have a doubling time of 20 minutes. So overnight, you go from one E. coli to billions. And it doesn't take any time to make a huge, huge quantity of DNA. So it's a lot easier to store. Obviously, it's stable at body temperature and room temperature and everything else. And it's a lot easier to use. It's a really interesting technology, but it's never been FDA approved, except for one that's used for racehorses against West Nile virus, I think. And so I think it's important from a security standpoint for like global biosecurity to have lots of people all over the place who can just, at need, make a vaccine. And it may not be a great vaccine, but it works, or at least maybe it works. and you never have to handle the virus. So that means the risk of me mishandling something and then it escapes from my lab is 0. So it creates a situation where you have a distributed network of possibilities that I think creates a sort of fundamental robustness that we don't have. And I think that's something we need.